differential expression analysis. How to do multi-factor differential expression analysis on SciDAP in easy way. To begin with, let's just get some basic information on when do we need to do differential expression analysis. So let's say we have multiple RNA-Seq samples and we want to see which genes are differentially expressed between them. And to do that, we usually start with the RNA-Seq analysis. We map all of our reads on the reference genome, then we calculate gene expression. We repeat this for multiple samples, and then we run differential expression analysis to see which genes are different between the groups of samples. And in order to do that, on SciDAP, we have several pipelines. We have 4 different versions of RNA-Seq pipeline that will calculate gene expression for either single read or parent data, and it can do it in a strand-specific or not strand-specific way. And then, using the results of these pipelines, we can run either DESeq pairwise analysis, which is our old workflow, and then 2 of the other workflows provide a better way to run differential expression analysis, allowing to have more complex design of your experiment. And I will describe how to use these 2 workflows. Previously, the first version of these workflows was developed by Valera from our lab, and I refactored them and made them more convenient to use in SciDAP. And the SciDAP is the platform where we can run any analysis, either written by us or written by somebody else, as long as they are written in the form of the CWL workflow. And CWL is just a common standard for describing your workflows. Inside each of these workflows, we have multiple steps. Each step is either a separate script, a separate tool. And in case of our DESeq pipeline, because the package is written in R, so I wrote these scripts in R, then put them inside Docker, and then put inside CWL, and eventually we can run them inside SciDAP. The reason we put everything in Docker is to guarantee reproducibility between the runs, because Docker is like a virtual machine with a pre-installed software. When we deal with the differential expression analysis, we have major problems to solve. Luckily, most of them can be solved by DESeq package, and that's the reason why we use it. And the major problems in differential expression analysis is that we have non-normal data distribution of our reads in genes. We have variable sequencing depth between the samples. We can also have certain genes that have very low number of counts. And we also need to make sure that the results, that we can run some statistic tests for the results to guarantee that the results are significant. But the other problem in the differential expression analysis, which is a responsibility of the person who analyzes the data, is to make sure that that person understands how to describe the data set in a way that DESeq will understand and provide the correct result. And here I just displayed what we usually use as input for DESeq. Most of the times it's just metrics with the genes and then the raw read counts. And inside DESeq, when we load this data, it's our responsibility to describe how the samples relate to each other, whether certain factors, and it can be treated, and for example, treatment, genotype, cell type, how these factors depend on each other. And then, based on all of these, how to extract the proper contrast. For example, treated versus untreated, or day 1 versus day 2, or maybe we want to see treated versus untreated in a day 1. And all of this is what we are trying to solve in an easier way using SciDAP. And to do that, we developed 2 of the pipelines. First is called DESeq-LRT Step 1, and then DESeq-LRT Step 2. And here I will explain first what are the inputs of the first pipeline. The main reason to have this pipeline is to run LRT test, which will give us all of the genes that are changed due to certain condition that we are interested in. It can be, for example, treatment, cell type, or some interaction between them. And also it provides a table of all of the pairwise contrasts that we can automatically generate based on the metadata. So I will go over all of the inputs and explain how each of them is used in our pipeline. The first input is array of the RNA-Seq analysis that we run on SciDAP. So each of these analyses is a separate sample. Then we define what kind of expression we want to use, either grouped by gene, grouped by isoform, or grouped by TSS. The most common is grouped by gene. Then, because we don't want to run the comparison having a lot of zeros, we can filter out the genes that are not expressed. And for that, we have a minimum RPKM threshold. Usually it is 3. That means we will remove all of the genes that are not that do not have more than, let's say, 3 RPKM among all of the samples. The reason to do this, because we don't want to compare one tiny number to even smaller number, have a very big log fold change, but in fact the actual number of reads was very small. So that's why we have this group. Then there is a group of parameters where we describe our model. And because this workflow runs LRT test, we have the design formula and reduced formula. And also we have a file where we describe how we can group our samples. I will show you later the example of this metadata file, but for now we can look at it as if it's a table that describes how our samples are grouped by treatment, cell type, genotype, or anything that we are interested in. The design formula usually looks like this, and it consists of what we call terms. Every term consists of variables, and the term can consist of one variable. So if we look at this, it's an example of the design formula. It's factor A plus some factor B plus some factor C that are independent of each other, and then plus interaction of factor A and B. That means that A and B, which can be, for example, treatment and genotype, interact with each other. For example, the treatment works differently for different genotypes. The reduced formula is something that excludes the term that we are interested in. Because when we run LRT test, we want to see the genes which expression pattern was dependent on the term that we removed. For example, we want to see all of the genes that were changed because of this interaction. So that's why we remove it from the reduced formula. And then there is a group of parameters that allow us to remove the batch effect. There are two ways how we can remove batch effect. One using the CUT&RUN package, which will adjust the counts before running DESeq, and another one is Lima package that will adjust the counts after we run DESeq, and it's mostly used to make the heat map look pretty. And here the parameter where we provide which column in our metadata defines our batch effect that we want to remove. It can be, for example, the day, or maybe the cell type in some situation. Then there is a parameter that is applied after we identified all of the differentially expressed genes. This is a filtering by p-adjusted to see only the significant genes. And then there is an optional parameter. When it is enabled, we will generate all of the pairwise contrast based on our metadata and design formula. And we did it just to simplify the next step for users. So a user can see a list of all of the available contrast, the number of all of the differentially expressed genes in each of the contrasts, and then just choose which of these contrasts he wants to see in a more detailed way. The next step is DESeq LRT step 2 pipeline. It takes the output of the first step. Then a user provides the name of the contrast that he wants to see in details. And then he also can provide several filtering thresholds such as p-adjusted, minimum log-fold change, and two of parameters that will make the result kind of more strict. So I didn't explain it on the previous slide, but this is exactly the same text that explains what the weld test is. So weld test is what we run in this pipeline in the second step. It just checks if the log-fold change for a particular comparison, or in terms of DESeq, it's called contrast, is significantly different from 0. And if we want to check if we want to compare, for example, is significantly different from a certain value, then there is a parameter in our workflow that will say that we want to compare, for example, 0.5 log-fold change instead of 0. And also, we can change what exactly weld test is checking. Either it is greater in the absolute value, or it's just greater, or it's less. Most of the times, these parameters should be used with their default values, but for some situations, when needed, the user can adjust them. And the output of this workflow is a heat map of all of the contrast, because we can provide several target contrasts. A heat map of all of the genes that were significantly differentially expressed in any of the contrasts. And then for each of the contrasts, we have a table of differentially expressed genes, volcano plot, and MA plot. And in order to explain it using a good example, I found a paper where they had very complex RNA-Seq analysis, and I tried to reproduce it, at least part of this paper. The reason why I selected this paper because they have several conditions how they group the samples. So I will try to briefly explain, without much of the biological details, what we have here and how I run this analysis. So they had 24 RNA-Seq samples. All of them can be grouped by tissue. This BAT tissue and SAT tissue, I have the explanation of these two types of tissue. Then within each of these tissue, we have samples that undergo DT treatment, which is diphtheria toxin, and no treatment. They just injected PBS, which is used as a vehicle for that treatment. And then each of these groups can be also divided by genotype. So in that type of the analysis, they wanted to see the effect of Treg depletion. So as I mentioned, we have two genotypes. Each of them was injected with this DT, and for DTR negative, it doesn't result in Treg depletion, but it gives some off-target effect of this toxin. For DTR positive, it depletes Treg cells and also have off-target effect. And for the samples where they just injected PBS, it neither depletes Treg cells nor have any off-target effects, but they injected it into two different genotypes. And if we want to see the effect of Treg depletion, the most kind of easiest way that we would probably select is just to have these 6 samples and then compare the samples that were injected with DT and the samples that were injected just with PBS. But that's not the right solution, because when we compare these samples, we see the effect not only of the Treg depletion, but also off-target effect of this toxin that was injected. If we select other 2 groups of samples, for example, mice that were injected with this DT, and here also injected with this DT, but one is DTR negative, another DTR positive, we would see the effect of Treg depletion and also the genotype-related differences, because we have 2 different genotypes. And if we compare these 2 groups of samples, we will see only the genotype-related differences. So how would we get only the genes that were changed because of the Treg depletion? And in our pipeline, the way how I was able to do it is to run LRT test with this design formula and with this reduced formula. So this design formula shows that we want to make DESeq to estimate all of the changes caused by treatment, tissue, genotype, interaction between treatment and tissue, tissue and genotype, treatment and genotype, and all three between them. And the reduced formula excludes these two terms. That means we want to see all of the genes which expression pattern was changed due to these two terms. This looks a bit difficult. Why exactly I selected this formula and why exactly this term answers the question. And in order to explain it, we will use exactly the same data set, but we will go from a very simple metadata to more complex, and I will explain what happens in DESeq on every step when we make the design and metadata more and more complex. So let's say we have 24 samples that are grouped only by treatment. So we have two groups, samples with DT injection and samples with PBS injection. When we run LRT test, the design formula includes only treatment. The reduced formula includes nothing. Then the results of all of the differentially expressed genes we want to filter by p-adjusted 0.1. So on this heat map, we see all of the genes that changed due to the difference between the design and reduced formula. And we have
using the log2 fold change also with the likelihood ratio test?
No, log fold change is applied for the table of the pairwise contrast. For LRT, as far as I understand, there is no way to interpret the log fold change properly, unless we run bulk test on the interaction term, and then we can interpret log fold change. So, but because our pipeline does two things at once, first thing what it does, it runs LRT test, and the second, it generates all of the possible contrasts, which then are filtered by both p-adjusted and log fold change. So these 52 genes, they were filtered by log fold change and p-adjusted. So this is the table of all pairwise contrasts that we were able to get based on the metadata that includes only one column and the design formula that includes only one term. So here, this is the result of the LRT test. Need to go there. And this table is the result of all of the pairwise contrasts that we were able to identify. So as we can see here, based on this metadata and this design formula, we can get two contrasts, DT versus PBS on average, and PBS versus DT also on average. Both of these contrasts are identical. The only difference will be in the sign of the log fold change. And we can see that because our design formula didn't have any interaction, here we have average and average. We estimated this main effect, which is treatment for DT versus PBS, and there is no interaction, and we got only 52 genes. This scheme shows how DESeq models it. So every dot here is an average expression of a certain gene per group. So this a certain gene from all of the samples that belong to the PBS group. This is a certain gene from all of the samples that belong to the DT group. In DESeq, one level is defined as a reference. In this case, it's PBS. And then it estimates the difference from this base level to the DT. So this A is what we see here, and the contrast C2 consists of only one main effect. So that's what's happening inside DESeq when we provide metadata with one column and design formula includes only one term. Now let's make it more complex. We will add one more term, tissue. In this case, we will first run LRT test, which will show us all of the genes that are different because of the tissue. Because you see, reduced has only treatment, design formula has treatment plus tissue. So all of these genes, and we can clearly see the pattern, are changed only because of the tissue. And this is the output of the LRT test. This is a table of all of the contrasts. I showed only two of them. We have four, but because they're the same, the only difference is the sign of the log fold change. I showed only two of them. This is the contrast that we looked in the previous slide. Now it has more genes, it also shows the changes between the DT samples and PBS on average because we don't have any interaction. But also, DESeq estimates another change, which is caused by tissue. So we have another contrast, which shows us the difference between BAT tissue and SAT tissue on average. So here we can see what happens inside DESeq when we have these two columns and this design formula. So as on previous slide, every dot is an average expression of a certain gene from the samples that belong to a certain group. So we have 4 groups, 4 dots. This A is what we had on the previous slide, the main effect treatment DT versus PBS. Exactly the same change is for the BAT tissue because we don't have any interaction, so DESeq assumes that the changes caused by treatment are exactly the same for both of our tissues. And this change is the change between the tissue. So this B is here, and this contrast C4 consists of only main effect tissue, BAT versus SAT. Now we try to make it even more complex. We will add the genotype. So now we have 3 columns. We added genotype. Now we have 8 groups. The LRT test will show us all of the genes that are changed because of the genotype. And we can see that the expression shows the pattern grouped by genotype. This is the list of all of the possible contrasts. Again, I excluded those contrasts that are repeated, just different directions. So we have 3 contrasts. This is the contrast that we looked before. Now it has even more genes. This is our treatment, DT versus PBS on average, because no interaction. Then we have this contrast that we also had before, and one more contrast that shows us the difference between the DTR positive and DTR negative. On the picture, this A, and here A are exactly the same for all of the tissues and both of the genotypes because no interaction. This what we had before. And this, another contrast that we see here, is the change caused by the genotype. So this thing is the difference caused DTR positive versus DTR negative. But what if our genotype and treatment interact between each other? And that's what we expect because we know that the treatment, DT treatment, should work differently in the DTR positive and DTR negative genotypes. So we will add this interaction into our design formula and see how DESeq will try to model these changes. I didn't show here the heat map, but the idea will be the same. We will see all of the genes which are changed due to this interaction. But what is more interesting to see is the table of all possible contrasts and this plot that shows what exactly is modeled by DESeq. So the first thing what we can see is that the tissue didn't interact with anything, so we still have average expression in BAT tissue versus average expression in SAT tissue, and it consists only of the main effect. There is no interaction. But because we have interaction between the treatment and genotype, now this contrast that used to be 1 is split into 2. We have DT versus PBS for DTR negative genotype and DT versus PBS for DTR positive genotype. The first one consists only of main effect. The second consists of main effect plus interaction effect. And the same is for the contrast DTR positive versus DTR negative. It is also split into 2 groups for only samples that were PBS injected on and only samples that were injected with the DT. And also here is the main effect. And this doesn't have any interaction because it's the base level. And here is the main effect plus the interaction. So if you remember, on the very first slide, I showed 3 different comparisons, and I told that we want to see the effect of only the Treg depletion. And if we look at these 2 contrasts, we see that this comparison, DT versus PBS for DTR negative, it shows the off-target effect of DT injection, because in DTR negative there is no any Treg depletion. But this comparison includes both the effect of Treg depletion because we injected DTR positive samples, plus the off-target effect of DT injection. If you look at these main effects and interaction effect, we see that to estimate this contrast, C9, DESeq sum up the contrast of the main the changes caused by the main effect plus this interaction effect, which is exactly what we are interested in, because this is the effect of the Treg depletion. So here I explained exactly the same what I just told, but in a slightly different ways, slightly different words. So this interaction, it measures how much the effect of DT treatment changes when the mouse was not DTR negative but DTR positive. Or in other words, does DT affect DTR positive mice differently than DTR negative mice? And this is exactly the biological effect that we were trying to answer. If you look on this plot, we will see that the only contrast that remained the same between DTR negative and DTR positive is what is caused by tissue. So this B here is exactly the same as here, but all of the other contrasts are now calculated differently. So on the base level, this difference, DT versus PBS A, in DTR positive genotype will be bigger. So it will include the change from here plus this interaction. The same will be for BAT tissue. We will have the change A plus this interaction. And this C5 contrast that shows us the difference between DTR positive and DTR negative genotype after DT injection will also consist of two effects. The main effect will be C, which shows us the difference caused by the genotype, plus the interaction effect, how the treatment worked differently in DTR positive mice. And this is how we analyze it when we also assume that there is an interaction between tissue and treatment, tissue and genotype, and all three of them together. So in this case, this heat map shows us all of the genes that were changed because of these two terms. And these two terms shows us, allows us to see the genes that respond differently to DT versus PBS, depending on whether the mouse was DTR positive or DTR negative, and possibly with an extra effect of the tissue. Or in other words, it will show us all of the genes that are associated with Treg depletion effect in any tissue-specific modulation of that effect, in any possible way, because we don't know if the treatment and tissue actually interact, but we asked DESeq to model that as well. And if we looked at the table of all possible contrasts, now we have 4 contrasts to show us DT versus PBS for SAT tissue in DTR negative genotype, the same for DTR positive genotype. Then we have DT versus PBS in BAT tissue DTR negative genotype, and the same for DTR positive genotype. So we started from just 49 genes that show us on average the difference caused by DT injection, but now we have 4 different contrasts. Each of them shows us how many genes were changed because of within a specific subgroup. And I checked if these genes actually involved in the processes that are described in the paper, and this is what I received after asking Microsoft Copilot, and it looks like most of them are involved in Treg depletion, and even some of them are described in the paper that I used as an example. If we want to look at the particular contrast, we can run DESeq2 step 2, and then, for example, provide the contrast name C12. Let's say we want to see the effect of Treg depletion and genotype-related differences in this specific tissue. So when we run the workflow, DESeq2 step 2, we can put the name of the contrast which we want to calculate. And it will give us a heat map, the values for log-fold change and p-adjusted for this particular contrast. And if we sort by log-fold change, we see that we were interested in this contrast between these samples, and indeed we see the pattern of the change between these two groups of samples. And at the same time, we see how these genes are changing in all of the other samples. This workflow allows us also to provide several contrasts, and then the heat map will include all of the genes that significantly changed in at least one of the contrasts. And also for each of the contrasts, we can show the volcano plot, MA plot, provide the TSV file with all of the genes, and so on. And here I will show you how I run it on SciDAP. So this is the first step. It starts from selecting the pipeline. Then we type the name. We select the samples, RNA-Seq samples, from which we will get the gene expression. In this case, it was 24 samples. Then we select how we want to group the expression, by gene, by isoform, or by common TSS. This is the field where we describe the design formula, and this is where we describe reduced formula. Then this input is optional. In this case, I wanted to correct by tissue to make the heat map look prettier, because otherwise on the heat map, we will only see the difference caused by tissue. Then there was filtering by PKM, filtering by p-adjusted, and filtering by minimum log-fold change for all of the possible weld tests. And this is the file that describes how samples are related to each other, the metadata file. So the first column includes the name of the samples that we selected, and all of the other columns can have arbitrary names, but you should use the same names in the design and reduced formula, so DESeq know how the samples are grouped. Then on the advanced tab, you can provide some additional options which alternative hypotheses will be tested in the weld test. You can also cluster the results by row or by column. You can select different distance metrics for clustering by row and by column. You can select how many levels of clustering you want and how many children in each of the level you want to have in the hierarchical clustering. Then when you save this sample, it will take, depending on the number of samples, a certain amount of time, and depending also on the complexity of your metadata. And these are the results of this experiment. You will see several tabs. The first tab will show you the LRT result. The second will show the weld test result. You can also see interactive MDS plot, which is similar to PCA, the heat map of all normalized read counts, a summary, which shows the metadata that you provided with the levels that you had, the number of genes that were selected for analysis after we applied minimum RPKM filtering, which design formula you used, which reduced formula, if you selected any of the batch correction method, how many genes you get after applying filtering by p-adjusted, and how many pairwise contrasts were calculated. In this table, you see all of the genes that were with a p-value calculated by LRT test. Also, the number of the clusters. NA means that that gene was filtered out by p-adjusted, but in this TSV file, you have all of them. The weld test shows you all of the possible pairwise contrast. And on the files tab, you can download the raw files for all of the inputs and outputs. Here we can see the MDS plot to see how the samples are grouped. You can color and change the shape based on any of your metadata column to see whether you have some batch effect that you want to remove. Then you have a heat map of all of the normalized read counts for the genes that are significantly differentially expressed. You can optionally add the clustering results, the names of the genes, calculated p-adjusted values, how the samples were grouped. And as you can see, we have a clear pattern of the genes that changed within the samples that were injected by DT between the DTR positive and DTR negative. And here we will see how to run the second step of this analysis. So first we select the type of the workflow, then we type the name of this workflow. It can be arbitrary. We select the first step, the result of the first step. We provide the name of the contrast that we are interested in, the filtering threshold for p-adjusted and log-fold change. On the advanced tab, we can choose whether this log-fold change threshold will be used in the alternative hypothesis testing and what the alternative hypothesis. Also, we can optionally cluster the result, provide different distance metrics for clustering, and also the number of levels and number of children in each of the level. When we save this sample, depending on the number of the contrasts that you selected, it may take some time, maybe an hour or two, to produce the results, and the results will look like this. So you have several tabs. On the first tab, you have just volcano plots, then wild test results, and all of the raw files. On the quick view, you have links to interactively see volcano, MA plot, and heat map. You also have the results summary. It is based on the inputs that you also provided in the step 1. So the metadata, the number of genes used, what was the experimental design, and then how many contrasts you selected and how many genes were identified as significant for each of these contrasts. On the volcano plot tab, you see the static images of all of the volcano plots. We have interactive volcano plots, so probably in the next version, these PNG files will be removed. The table shows all of the genes that are changed in any of the target contrasts. For each of these genes, you have several columns, p-adjusted, base mean, log-fold change, and the prefix of the column shows from which contrast it came from. And you also have the clustering results. And here you can download all of the raw files. And if you go on the Overview tab, click on the volcano plot, you will see interactive volcano plots for every of your target contrasts. You can change the colors. You can see which dot correspond to which of the gene. You can change the filtering threshold. You can export it as a PNG or PDF file. The same is for MA plot. Also, you can choose any of your target contrasts. You can adjust the colors, save it as a picture. And also, you can see the heat map of all of the genes that were identified as significant in at least one of the contrasts. And in this case, we run clustering because we have several target contrasts. If it was only one, probably I would just order them by log-fold change. And from this Morpheus heat map, you can also download a PDF version or PNG file. So this was an example of how we run differential expression analysis. The same way, I'm going to add a new set of pipelines to run differential accessibility analysis. It will also give us an option to see the table of all pairwise contrasts and then choose which contrast we are interested in. And well, many thanks to all of you who came to listen to my talk, and also thank you for all past and present members of the Varsky Lab, and also Microsoft Copilot for correcting my grammar and adding a proper explanation for something that I wasn't able to make in my head in a proper way. And this is the data set. This is the paper from where I took the data sets. Thank you.
Michael, this was a really good presentation, and I think it really highlights how well, or how these DESeq2 GLMs are, getting to the values that they do. I think I have an idea and a question for you, and maybe maybe the idea would be, do you have any plans to implement support for results or quantifications that are coming out of pseudo-aligners, such as Salmon or Callisto? And then I guess I'll ask the other question in a second. So I guess the first was, do you have any plans for something like that?
Well, it will depend whether we will have any project that will require that type of alignment. Currently, we use star for alignment, so I haven't tested. Oh, I think Artem has the answer.
I can answer this, and yes, in some cases, some pipelines use Callisto for pseudo alignment to some of the genomes, and those inputs can be used as inputs into DESeq. At least in the previous version of DESeq. I'm not sure if Misha added it to this one.
I can add that I haven't tested the output from pseudo alignment in DESeq. Most probably, we will need to use some other parameters in DESeq, because I know, for example, that on the manual page of DESeq, they even explain how to run it for single cell data. Most probably, they have some advices how to do it with the pseudo alignment results. But eventually, it will depend whether we will have a project that requires it.
I don't think it's
only reason I bring that one up for you is because it would probably be a fairly easy win to add some more functionality for people that are quantifying their data in a slightly different way, and it would be very easy to import. So there's a package called TX-Import that will essentially convert those data sets to raw counts that can be used through DESeq2. Very, very simple. So that might just be something that would be worthwhile considering. I think the other question I have would really be around, I like the idea of visualizing corrected counts from lemma, which I'm guessing is the remove batch effects function, where you've passed in a certain design.
Yes.
Do you ever run a pipeline the same way with and without that visualization, just to ensure that it's not a single sample that's driving that effect? So I've seen some cases where it will look very good after you've removed that batch effect. From a heat map perspective, everything looks nice and consistent. But if you take it back to the original normalized data, you'll get these streaks where it'll be a single sample that has driven that gene being called as significant. So do you ever do that, or is there how do you think that you could approach something like that?
Well, most probably I would just build the PCA to see whether how the samples are grouped, and if there are any samples that are far away from all of the other groups. I didn't have this situation with this particular data set. I had similar situation running DiffBind, which internally runs DESeq. And for that, I had to remove some of the samples, and then the results looked much better. As for using lemma, I believe this is kind of the safest way, because it only changes the normalized read counts that are used for heat map after we already run DESeq. I would be more careful running CUT&RUN, because it will change the values that go to DESeq. And in that case, we can kind of make it worse.
Yeah, thank you.
Okay. That's it. Thank you.